Got to get the clicker. Good morning, everyone. I'm just going to do a couple of case studies, and there's going to be a few take-home messages that I would like you to understand. And if you have any questions, of course, they'll be at the end when we do the panel. Okay? And as we heard earlier, the facility and the process review is together. And again, it's also part of the team, right? But our learning objective for this is to provide insight into how we review the chemistry manufacturing controls, how we assess what you submit to us. Specifically, I'll be talking about risk assessment, development data, and facilities and equipment. First, we have a poll question. And this is mostly for our people at home, but I hope you participate in the room too. What is the most challenging aspect of creating your chemistry manufacturing controls section of your application? So here in the room, who says development data? Show of hands. Oh, that's the easy part? Okay. How about the risk assessment? Is that hard for anybody or more difficult than other parts? So I see some hands about risk assessment. I even see somebody way back in the back raise their hands. What about the critical process parameters? I see a few hands on that. And then there's acceptance criteria. What do we think? Okay. And I see the votes are just about in, but it looks like risk assessment is running away with it. So hopefully there's a few people in the room that have concerns about risk assessment, but they're just a little shy about saying it. So with respect to your CMC, a big part of it is your risk assessment. A big part of it is your development data, and a big part of it is your facilities, right? When you're doing your CMC, do you think about all the facilities that are involved when you're writing it? Yes, no, yeah, that's good. Well, the risk assessment is to find out if there's any risk. We need to identify that risk. You know, in a lot of movies, they say, what's the worst that could happen? But when they say that, they're usually meaning nothing could go wrong. And of course, in a movie, everything goes wrong after somebody says, what's the worst that could happen, right? Well, with a risk assessment, we want to ask that question. What could go wrong? And if it goes wrong, how severe is it going to go wrong, right? Also, what is the probability that it's going to go wrong? Those are the things we want to think about when we're doing our risk assessment. So when we find that risk, we got to figure out how we're going to mitigate it, right? So now we're going to do all these development studies, right, to get a handle on what we're going to do and how we're going to mitigate the risk. Now, there may be some unit operations that you don't think there's any risk, and that may be true. And hopefully you will say that. You just won't leave it out, right? You will say that this unit operation is minimal risk because X, Y, Z. Now, with the facilities, when we're looking at your risk assessment, when we look at facilities, we're not just looking at that facility, we're looking at the history of that facility. So when I say that, I mean we're going to be looking at your past inspections. And if we see something in the past inspection that looks like that could be a problem for this application, right? Well, we will look at everything that FDA has on the facility, including, are there field alerts out there? Any quality defect signals? We check all of that. Do you think about that when you're writing your CMC? Do you think about the history that FDA has on your facilities? Everybody's looking at me like, why is she asking me that? And so, and with your development studies, we look close at your development studies because that tells us how you arrive at what you're going to do. And that tells us whether you're going to need an in-process control or not, or if the in-process control is suitable for what it's supposed to, what risk it's supposed to mitigate. So all that is a part of your CMC. So my first case study is a lozenge, which we're treating as a prompt release tablet, because basically that's what it is. It has a low drug load. This particular drug product has two strengths, 2 milligrams and 4 milligrams. But the amount of drug substance is like 1% and 2%. So that's really low. And this is a dry blending direct compression, which seems kind of easy, right? We think that's easy. Especially if you've done it many times, it's really easy to do. You would rather do this than wet granulation any day of the week, right? Just one problem. You don't have a history doing it. This is a new profile at your facility, as Rocky said. That's a risk because they've never done dry blending. They've never done compression. We've never looked at their compression operations in a previous inspection. So that's something we would look at. So then we do an initial risk assessment. And as most of you, when you submit an application, you also do a risk assessment. We may or may not see the same thing when we do our risk assessment. We might agree with you, we might not. But for this lozenge, the unit operations that carried some risk were pre-lubrication, lubrication, and compression. And they're linked to the critical quality attributes for pre-lubrication. Is that me? Maybe it was me. Okay. And for the pre-lubrication and lubrication, it's assay and blend uniformity. And for the process risk, it was medium. And for the facility risk, it was high. And if you recall what our considerations were, it's a low drug load, no dry mix process experience, no tablet experience, but there's been no observations for the previous two inspections, and there have been no quality defects. Now with compression, the quality attributes are assay, content uniformity, and dissolution. And so the process risk here for this initial risk assessment was high. And the facility risk remains high because we have no history on these operations. So we always want to worry about what we're putting in, right? Garbage in, garbage out. Quality in, quality out, right? So we want to look at everything we're putting into the process to make sure that it has attributes that will not affect our drug product adversely. Would you agree with that? So for the drug substance, we have poor flowability. On this application, they did a lot of work with the drug substance. They looked at the compressibility index. They looked at the Hosner's ratio. They did a bunch of studies to prove that the flowability was poor. It's also hygroscopic. So since we know that it's hygroscopic, as a process facilities, we want to make sure that they're capable of controlling and monitoring, or I should say that reverse, monitoring and controlling the humidity so there won't be an effect on the drug product. And it's also controlled for particle size distribution, which if you don't want to do any sizing of the particles, that's always a good thing. If you're controlling it before it goes in, that's a good thing. However, there's an excipient that's 85% of the tablet. That excipient happens to be mannitol. Mannitol has good flowability, and it's also controlled for particle size. So now we have pretty good assurance that when we make the product, we're not going to have any flow issues in the process. So we looked at their blend development. The good thing about their blend development was they defined their critical process parameters. That's always a plus. We like to see that. And for something as simple as blending, because you're just tumbling the thing, right? It's diffusive. And the only thing they looked at was mixing speed and mixing time. But they also included a percent occupancy. We see applications on a regular basis that don't include that information. That's not to say that they didn't do it. They just didn't submit it. But when it's not submitted, that causes us questions. And our questions come in the form of information requests, right? So if the fact they included it was one less question that we had to ask. So we're very happy about what they did so far. But then there was a few things that could have did better. They had all this development data, but the data was not summarized adequately. So they had passing lots interspersed with failing lots. It wasn't necessarily chronological in the order that they made the batches. They intermixed both strengths in the development data, which makes it a little harder to wade through it. And then you find yourself saying, wait, what? I'm a little confused. And so if we're confused, again, we're going to ask the question. And so for this application, we asked them, could you please summarize the data? And we asked them to do it chronologically with the batches. And that's the order they were made. And having said that, if you put the first development batch and then the last development batch, but we can tell by the numbers that there's a whole lot of batches in between, and you've made changes from here to here, you might need to submit more of the batches actually to tell your story about how you got to where you were at the end. So it's better to submit all of your development batches in order. And along with those, now I'm not saying if you got 1,000 batches, you submit all 1,000. But you submit enough to tell your story. So you should have your batches chronologically. You should put whatever you're testing for to prove this step. You should put what the condition was. And you should also put, did it pass or fail, right? And separate the strengths, because one thing that might be going on in one strength may not be going on in the other. And it's a lot easier if they're apart, right? And also with your exhibit batches, a lot of people summarize the final product for the exhibit batches. We get a nice table, all 3 exhibit batches together. It's also nice to do that with the in-process testing results. Some people do it, some people don't. It makes our lives a little easier. I don't know why that's happening. Okay, must be me. So anyway, the more you summarize the data, it's easy for us to get through it. And also we noticed that the blend uniformity data wasn't robust. And that could be because, we're down 1-2% of the product load, right? So the mixing may not be as good as it needs to be. But I'll come back to that in a second. Compression. The critical parameters were defined again. We always like that. And for here, it's the speed, pre-compression. I didn't do it that time. I should what? Oh, okay. Okay. So a lot of times pre-compression is not defined because it doesn't have to be a critical parameter. But it's nice to have. We're happy to see that. And they did content uniformity via stratified sampling. They probably should have did stratified sampling with the blend uniformity as well. It would have given them a little more, I think it would have helped to be more robust. But they did it for the content uniformity. And they used 905, USP 905 for their criteria. Now, as far as this compression, They came up with some settings that they were going to use, but then those process parameters were adjusted for the exhibit batches. After they did all that development data, they had to adjust during the exhibit batches. That leads to questions. And then the pre-compression force was not sufficient to always produce passing tablets. So that's really interesting, right? But when they did the development, they did a range of like 5 to 20 kilonewtons, right? But when they set their acceptance criteria, they did it 3 plus or minus 2. They kind of painted themselves into a corner. But since they had all the development data, we knew it wasn't so much an issue with supporting it. It's just that they narrowed it for some reason. And so we recommended that you open it up a little bit. So when you changed your exhibit batch, you were still within your development data, but it's not what you concluded from your development data. So this facility had a good inspectional history. They resolved any deficiencies they've had in the past. There have been no quality signals, but there's no experience with proposed manufacturing operations. Do you think we asked for a PAI? I see some heads nodding. Yes, we did. But it turned out the inspection went very well. So the lessons learned here: organize and summarize your development data. I can't say that enough times. And just to give you a flip side of that, I've had applications where I have tables and tables of data, right? One paragraph of words. So now they've gone too far the other way. And so when you have all this data, you need to put that data into context. The context is what makes the data show that it actually works, that your conclusions are actually in line with the data. The development data should support your controls. And we saw that the development data was wider than what they decided to pick, but they were able to go back and change it after we sent the information requests. But you also want to make sure, if you do go back and change your criteria, that your exhibit batches actually pass those criteria. And if some things had happened along the way, as we know they always do from time to time, if there are deviations and failure investigations, include them. It's not a requirement to include them, but if it's what caused you to change your development into another direction, it'd be nice to include those. And if you allude to deviations in the exhibit batch records, because we go through all the batch records, it'd be a nice idea to go ahead and include them, especially if you alluded to them. The second one is a capsule. And again, capsules, they're pretty easy, right? This one is prompt release, just like the tablet. Except now we're drug coating sugar spheres. And this facility has several applications where they've been approved for capsules. So they have some experience making capsules. And the drug load here is 3% for all 3 strengths. And I believe the strengths were 2, 4, and 6 milligrams. So this is initial risk assessment here. And the unit operations that were included were the drug solution mixing, the drug coating, drying, and encapsulation. The drug solution mixing was low. The facility risk, again, is high because probably the quality defects and their inspection history was not, let's say, stellar. So the process risk is low. The drug coating is high for both counts. And there are going to be some process parameters with the drug coating. Then we have drying, and then we have encapsulation. And so, again, there may be some process parameters that may need to be worked out. So I said that this is sugar sugar spears are going to be coated with the drug. Well, the drug's going to be solubilized in water, the drug substance. So what do you think a critical material attribute is? It's going to be put in water. Do we want it to dissolve in the water? Yeah, we do. Okay. So it's the solubility, and it's highly soluble in water. So making the drug solution is indeed low risk. So in the drug coating development, again, they came up with their critical process parameters, and they used design of experiments. And they also used the quality by design approach, which you've heard from earlier today. It does make things a little easier. And so we're looking at product temperature, airflow, atomization, air pressure, and spray rate. Those are the critical parameters that they looked at during their COE. And they had in-process controls, which is always a good thing. What they could have done better was the critical process parameter ranges used in the exhibit batches weren't the ones that were developed during the development data. And then they had a spray rate during the process going from 30, 40 to 300, which is kind of wide, right? So again, that led us to ask the question. And so what they did was they put the drawing in 2 steps. So the first part for 60 minutes, because that's when it was changing after 60 minutes. The first 60 minutes was like 40 to 100. Then the second 60 minutes was like 100 to 300. And they did another couple of batches to show that it worked. Encapsulation. You know, with today's machines, it's really not that hard. They have dosimeters. Everything goes pretty smoothly. And there's one critical parameter, and that would be the machine speed. And there are a bunch of in-process controls. And the only reason you have the average weight of the empty capsule is because they're going to subtract that when they do all their other calculations. So they did a great job with encapsulation. So they had a recent inspection that covered high operations, the high-risk operations, but there were those pesky quality defect signals in the form of field alert reports. However, those field alert reports related to tablets, and they were tablet mix-ups. So it didn't necessarily didn't add any risk to the capsule, because when they made capsules, everything always went smoothly. So did we recommend a PAI? We got a 50-50 shot at this. Yes, I see one yes. See another yes. We actually didn't, because the high-risk unit operation was covered during a very recent inspection just 1 year prior. And even though they had issues in the past, it looked like they had all been worked out. So that's always a plus. The lessons learned, the exhibit batch process parameters should match the development conclusions. And the reason I have is for information only, a data or a control. They were controlling the coding process with an LOD, but they had a F for information only for weight gain, which normally we don't do for coding drug spears, but they did. But the question is, were they making any decisions off that data? And it turns out they weren't. So in that case, it truly was for information only. But if you have something that says information only, and then you turn around and you're making decisions off of it, then it's really not for information only. And for whatever you do, provide justification. That is so important, that we understand what you're thinking, what your rationale was. So when you're doing these things, we want you to consider your operation holistically, as Mahesh said. We're looking at this part of your process, but we're part of a team. If we see issues with the drug substance or questions, we're going to huddle with the drug substance reviewer. If we see an in-process parameter that doesn't fit, actually a acceptance criteria that doesn't fit with the final drug product specifications, we're going to talk to the drug product reviewer about that. So you want to look at everything from beginning to end, cradle to grave. You want to make sure that your decisions are science-based. You don't want to do the equivalent of where you're saying, well, because I said so. That's why. But it may be you actually have a lot of experience. And if you say we have a lot of experience, then we know why you feel the way that you feel. And again, can't say it enough, summarize your development data. These are team members that helped me put this together. Challenge question. What are the 3 fundamental questions used to define risk for risk assessment purposes? What can happen? Right? Right. What might go wrong? What is the probability that it could go wrong? And what is the severity of it going wrong? What is the aim of pharmaceutical development? Oh, wow. We want to make sure that you're making quality products, right? We want you to design a quality product, and you want to come up with a process that consistently delivers a safe and efficacious product to the patients every time. That's why development is so important. That's why there's Q8, right? There's Q9 for the risk assessment. There's Q10 for the system, the quality systems. Thank you.